So welcome everyone. Today we are hosting Professor Rafael Yuste from Columbia University. Rafael started his academic career in medicine at the University of Autónoma in Madrid, Spain. He then did his PhD at Rockefeller University with Thorsten Wiesel and Lawrence Katz, and then his postdoc training at the Bell Laboratories with David Tank. Today, Rafa is a professor at the Columbia University, a co-director of the Kavli Institute of Columbia University, and a director of the Neurotechnology Center at Columbia. Rafa holds a long list of awards and prizes. I'll mention just a few of them. The NIH Director Pioneer Award, the Telva Science Prize, and the Van Ever Bush Faculty Award. Rafa studies a neural circuit with the goal of trying to decipher the relation between activity of neuronal assemblies and behavior and mental states, and he uses electrophysiology and optical methods to record and activate neurons within circuits. He does this in cortical circuits, but also in an organism with a simple nerve system, the Hydra vulgaris, which he uses to try and decipher the basic and hopefully universal principles of the neuronal code. So before we start, please make sure that you are all in mute mode. I'd like to remind all of us that we like questions at the end of the talk, but we of course allow questions during the talk as well. So without further ado, I welcome Professor Rafa Yuste.
Thank you, David. Good afternoon, everyone. Greetings from New York City, where it's snowing, and we're in the middle of a snowstorm. So I have to give this talk from my living room. I couldn't go to the lab today. And the talk I wanted to tell you about starts with magnetism. So this all starts with magnets. It turns out that something very interesting happens with magnets, which is that if you take a magnet apart into the individual atoms, the atoms are not magnetic. But if you put them together, something magic happens and the system becomes magnetic. I'm talking specifically about ferromagnetism. And this caused a huge headaches to the physicists 100 years ago, because they said, well, how can you get something out of nothing?
Rafa, we cannot see your screen.
Oh boy. How about now?
We see the screen.
We can see it.
No, we see the screen.
Everything
you can see the screen. Okay.
We see.
All right, you should see a picture of a magnet and a picture of Ernst Ising right here. Can you see my cursor, by the way? This is also important. Perfect.
Yes.
Anyway,
Yes.
So what I was saying is that magnetism is a classical example of what we call in science an emergent property. Okay, and emergent property is a property of a system of particles of units, which by definition is not present in the individual units. Okay, and is not magic. And this guy actually, Ernst Ising, was an undergraduate in Germany in the 1920s. And he was asked by his professor to solve magnetism, and he didn't know it was a difficult problem. And he solved it with this very simple idea that the emergent properties are generated by the interaction between particles. So they're not present in the individual particles, they're present in the interactions between the particles. And Ising proposed this concept of energy. Energy in physics, you could think of energy as the propensity that the system has to change. So you have a lot of energy, you can do change a lot, and very little energy, you can be very stable. And he defined a negative energy term, which depends on the spins of the individual atoms. And if they're coherent, these spins are going to be actually multiplied positively times what he calls the coupling coefficient, this J term, which is essentially the strength of the connection between these atoms. So if the spins are coherent, the connections are very strong, this term is very big, which means that the energy is very low, which means the system is very stable. And with this idea, the physicists actually understood for the first time magnetism. And this is a wonderful example of how an emergent property can actually be approached to by science. So why do I bring up magnetism? Because now, if you look at the nervous system, you have to realize, particularly if you look at the evolution of the nervous system, that it's built with an increasingly higher and higher astronomical high number of units. So it's estimated that we have about 86 billion neurons in our skull. And then these units are actually connected in also almost astronomical numbers. So it's possible that every one of the neurons in our cortex, for example, is connected to another 100,000 neurons. So, and this, if you look at how the nervous system increases in size and connectivity through evolution, you get the feeling that what nature is doing, it's playing the emergent property game. Because if you want to design a system with emergent properties, what do? Well, you build a system that has as many units as possible, and you connect them all as much as possible. So this is a strong suspicion that we don't understand the brain because we're dealing with an emergent system, a system that's actually playing the function of the brain is written on these emergent properties, which would be interaction, not between atoms, but between neurons. And these would be interactions in space and in time, and they would form units, just like in the magnetism case, these units, these spin glasses are actually territories where coherent spins generate strong magnetic forces. So there could be some equivalent, formally equivalent states in the brain where coherent states of activity would generate these fleeting emergent functional units. So now if you think like that and you look back at the history of neuroscience, you have to realize that for almost 100 years, neuroscientists have been anchoring their approach in what's known as the neuron doctrine, okay, which was stated by Causal, he got the Nobel Prize for this, and also by Sherrington, who also got another Nobel Prize for something for the same idea. And this means, by the way, and they call it a doctrine. It was not a hypothesis, it was a doctrine, religious belief. And the argument is that the unit of structure and function of the nervous system is the individual neuron. Both Causal and Sherrington were pioneers in terms of using methods to study individual neurons. Causal used the Golgi method that labels individual neuron in the middle of the brain parenchyma. And Sherrington was the first person to use electrodes to record the activity of individual neurons. So since then, neuroscientists have taken apart the brain, studying it neuron by neuron. And in terms of function, Sherrington and the school that he trained, of which I am actually a proud descendant, so he's my great-great-great-grandfather, scientifically, of course. What we've been doing is using electrodes recording the activity of one neuron in a brain of an animal, okay, and correlating the function of that neuron, the firing of that neuron, like in this drawing, with the behavior of the animal, or if it's a patient with the pathological state or mental state of the patient. And if you think about emergent properties, this is a little bit like trying to watch a movie in a TV if you look at individual pixels. So it doesn't matter for how long you look at a single pixel, you will never understand what's playing in the TV. No? Why? Because the images in the TV are emergent properties and by definition are not present in the individual pixels. So you never get it. You're never going to understand these emergent properties if you look at the system unit by unit. And these emergent properties of the TV are based on correlations in space, in time, and color of pixels. So they're not magic. They are right from the correlation of pixels, but you have to capture them all in order to see these emergent properties. No? So what I'm telling you is that neuroscientists, because of the neuron doctrine, have been doing the wrong experiment by trying to understand a system that could be emergent. In fact, it's been the mother of all emergent systems designed by evolution precisely to play that game by taking it apart one unit at a time. And by definition, you're never going to find it. So this is not an original idea. This was first suggested in my book by Laurent Thévenot, who was a disciple of Causal, who spent his whole career looking at studying these circuits of neurons in the forebrain and in the cortex these circuits that Causal had called the impenetrable jungles, where many investigators have lost themselves. And Lorente, like a good disciple, he spent his entire career doing the opposite of what he was told and getting lost in these impenetrable jungles. As his conclusion was that the connectivity of the brain, of the cortex in particular, was recurrent, was a feedback connectivity. So here you have one of his drawings in which you have this neuron that receives an input. This is all supposed to be excitatory inputs. And this neuron connects to this other neuron, which connects back to it. And this forms a loop, okay? And he saw loops everywhere. He called these loops change, reverberating change. And he argued that the point of the cortex, the point of the brain, is precisely to generate this reverberating change, which have a very special property, which is once you turn them on with your input, they're on independent of the world. They're like switches. They live in a state in which they're controlled by themselves. They don't depend on the outside. And this little detail has enormous importance. And by the way, this is the same idea that Alan Turing came up with this idea, this drawing here of one of his papers. Because with these feedback loops, this reverberating change, you can start building a system in which you have internal states, okay, which are independent of the world. And then you can use these states as symbols to represent the world, okay. This is also not an original idea. It was first proposed, I think, by Kant. He called these states Darstellungen, representations, and they were internally generated. So in a way, we're talking about having a vocabulary of internal states. We could have many of them in our head, and we use them as symbols to point to different things in the world, which means that instead of manipulating the physical world, we manipulate this model of the world, this symbolic structure. And this is the beginning of cognition. So the same idea from Lorente was proposed by Hebb. Hebb credits Lorente in his book, but he changed the name because every scientist who wants to leave your imprint, he wants to be the one whose name is remembered. So instead of using the word chain, he came up with the word assemblies. It's exactly the same idea. And he's one of his drawings from his book in which you have this graph in which the nodes of the graph are neurons and the arrows are connections. And he said that these he called assemblies would be essentially on internally driven, and they would be, it could be used, for example, to represent a memory. Hebb proposed something new besides Lorente, and it had to do with how synaptic plasticity formed these assemblies. He said that if you assume a synaptic plasticity rule, like the Hebbian rule, neurons fired together would wire together. He said it would be unavoidable not to have assemblies. In other words, if you have a system with recurrent connections and this type of synaptic rules, whether you want it or not, you're always going to end up with assemblies. The neurons will group into assemblies. They stick to each other, forming these groups through these synaptic connections. So then all of these ideas were very stimulating, but it was actually John Hopfield in a very inspiring paper in '82, where he provided mathematical formalism behind these internal states. And Hopfield came from physics, from condensed matter physics, and he actually took the Ising model exactly with the same equation. This is the Hopfield equation. If you recognize it's the same equation as the Ising equation, he defines this energy term, which is also negative, which depends on this case, instead of the spins of neighboring atoms, is the activity of neighboring neurons, of connected neurons, and this T term is the coupling coefficient of Ising, which now is actually the synaptic strength. So according to Hopfield, if you have a circuit which is recurrently connected, just like Lorente and Hebb have argued, this system is going to stick to these stable states, just like in magnetism. And he called these states attractors. because they would attract the activity of the network to that state. And he proposed that you can visualize these attractors, which of course have many dimensions. They have as many dimensions as neurons, but you can imagine them in 3D, in like a plot, like a contour plot, like this one here, where every point in this plot will correspond to the state of the network at any given time. So imagine that this point, all the neurons are on, and in this point, all the neurons are off, and all these intermediate points are all these intermediate states where some neurons are off, some neurons are on. And the third dimension, the depth here, is the energy. So these attractors are these little valleys where, if you think of neuronal activity in the brain as a ball that's rolling through this landscape, when it gets close to the attractor, it would drop automatically into the bottom of the attractor, and that would be state. That's stable or semi-stable, at least stable for a little bit of time. And he argued that these attractor states could be used to implement associative memory, or more generally, to implement a solution to a computation. This is a neural network model where the attractors would be the solution, let's say, to an optimization problem. And the ball rolling through the landscape is what Hopfield called the dynamics. That's the computation itself. Hopfield pointed out something that Hebb had also talked about, which is that this system has pattern completion. So what does it mean? That if you don't have to specify the system all the way down to the bottom of the attractor. You have to get the system close to the lip of this valley, and then automatically the dynamics of the system, because of this energy term, will attract the system to this lower energy state. So this is actually something that happens in memory, where you remember, you can recall a memory by only having partial information. Pattern completion is actually widespread phenomenon in behavior as well. Anyway, so I was lucky. I spent 5 years at Bell Labs in the same department as John Hopfield, and I had many times lunch with him. And I came from the, as you heard, from the Whistle lab. Sorry. So the Whistle lab, this is like Sherrington, single electrode people, neuron doctrine 101. So I had enormous discussions with Hopfield, arguments every time. And I remember one day I asked John, well, I mean, your model is very interesting, but how can we prove you wrong? Okay. You know, in science, to advance science, you have to falsify the model. You have to design a model that can be tested right or wrong. And John laughed, he laughed and said, well, Rafa, it's going to be pretty tricky because, first of all, if you have to measure every spike from every neuron so that you can see the entire attractor landscape, and then you have to move the ball from one place to the other. So you have to be able to turn on and off every neuron in the system. And if you can do that, then you can bring the ball close to the attractor basin and see and predict how it behaves. Or maybe it doesn't behave like that, and then you prove me wrong. Anyway, so I walked out of Bell Labs with those ideas in the back of my head. And I have to tell you that another person here that provides also another huge insight was your own Moshe Abeles, with a completely independent approach. As you know, he's a biophysicist, a physiologist, and thinking hard about the way that the cortex is connected with a lot of weak stochastic connections, and thinking hard about the threshold of the action potential, which becomes increasingly non-linear as you get close to the threshold, the probability of spiking. He proposed the idea that the cortex was built to generate synchronous states of activity. So these synchronous states in my book are just like attractors, it's just like chains from Lorente, it's just like assemblies. He called them chains, going back to Lorente. And he also added to the picture the idea that these states would essentially fire each other in a sequence. So this is a drawing from one of his reviews in which you have every one of these gray bars, this synchronous state, one of these little attractors, that would then fire the next attractor and the next attractor. And this is what he called the syn-fire chain. So he said that the system is designed to select for this type of activity. If you have activity which is not synchronized, it will die out because the synapses are weak, stochastic, and also because of this issue with the action potential threshold. And he provided some data using multi-electrode arrays. He and his co-workers, and I think some of you came from the ABLES lab, suggesting that there were actually these precise firing sequences in the cortex of awake behaving monkeys, which correlated with behavioral decisions and which had the property that their timing was actually very precise. And this is something you can understand if you think that you're just reading out a few neurons from this long chain. So these are all fascinating ideas, but how can you test them? So, okay, so this is an example of the piece of cortex that we look at. This is the primary visual cortex of a mouse. Every white dot is a neuron. Just to give you an example, the P1 in the mouse has about 180,000 neurons. So if we think that the work of the cortex is to build emergent states, going back to this comment from Hopfield, well, what we need to do is very easy to record every spike from every neuron, and then we see all these emergent states, like watching all the pixels in the TV screen. No, it's simple. How can you do that? Well, if you try putting electrodes there, this is not going to work out. No? You're going to turn the whole thing into a switch. So we need methods to record every spike from every neuron and to manipulate every spike in every neuron in a neural circuit. And this was exactly the idea that was proposed to President Obama that led to the US Brain Initiative. The US Brain Initiative is a 15-year-long project which involves currently 500 labs in the US and around the world, which is going to be funded totally with approximately $6 billion. And the purpose is to build methods. Methods for what? To do three things: to measure every action potential from every neuron in the brain circuit. And you can imagine starting with the small animals like cnidarians or worms. And as we build better methods, graduate to bigger and bigger animals, maybe measure the entire activity of all the cortex in a mammal, or the activity of every neuron in a piece of cortex of a patient, for example. The second goal is to develop methods to actually manipulate the activity so that we not only see, but we can actually get in with our little fingers and test different hypotheses of how the system works. And the third, which is equally important, is mathematical methods to analyze the system, because we're looking for emergent properties, and we probably have to invent new types of theory and math to discover these. These things are not easy. The reason why science has not dealt well with emergent properties is because they're very difficult to formalize. Now, this interaction between particles, particularly if they're nonlinear, and they're all nonlinear, is not so easy to capture a mathematic and to predict. I should tell you that the Brain Initiative has also inspired brain initiatives in other parts of the world, including China, Japan, Korea, Australia, Canada, and also in Europe. A similar project was launched at the same time. And all these brain initiatives are now banded together in an international brain initiative. This is which was formed in Canberra, Australia, where representatives from all the brain initiatives signed a document to work together. So this is a revolution in terms of methods building for neuroscience, and it's going well. There are all kinds of methods going on, but time will tell which is the methods are going to win the race. A little bit like what happened with the Human Genome Project, where the methods that ended up being used to sequence the genome were not the ones that were used at the beginning. So now let me turn and discuss to you how in our lab we're contributing to this type of question. How can we search for these emergent properties of neural circuit? And this starts when I was a grad student at the Rockefeller working with Larry Katz, who unfortunately died young and whose memory I honor here. He was my mentor. And we discovered by chance that you can use these calcium indicators, which are essentially fluorescent molecules that change optical properties when they bind calcium. And you can use these indicators to load neurons in brain tissue. So this image that I showed you earlier is actually a brain slice, a living brain slice. So every one of these dots is a neuron that's fluorescently labeled with these calcium indicators. So this was a chance discovery, I should say. No? But what made it even more interesting was to realize, also by chance, that when you image the fluorescence of calcium in these neurons, you see changes in fluorescence that correspond to action potentials. So this is illustrated here. You have a recording, intracellular recording with a patch electrode of a neuron as a function of time. These are the action potentials. And if you simultaneously measure the fluorescent intensity of the calcium indicator, you see these increases in intracellular free calcium that correspond to the opening of photosensitive calcium channels every time the neuron fires an action potential. Okay. So this signal, this calcium signal, is slower. It's a low pass version of the electrical signal because of the biophysics of the calcium. So that's a limitation. But the good thing is that you can measure it simultaneously in all the neurons in the field of view. So this is an example here. You have 500 neurons, primary visual cortex of a mouse. Every one of these gray dots is a neuron, and this is 10 minutes in the life of this piece of cortex. And whenever these neurons have an intracellular increase in intracellular free calcium, we've labeled them red. No? So again, this is a little corner of the TV of this primary visual cortex of the mouse, which is a little corner of the TV of the whole mouse brain, which has 100 million neurons. So we're only looking at 500 neurons, but in that little corner, we can see every neuron, okay? And we can start exploring if there are emergent properties. And this was helped also with another method that I was also lucky to participate in its development, and this is two-photon microscopy. I will not go into details of how it works, and this was invented by Winfried Denk and Watt Webb, and I was lucky to work with Denk when I was at Bell Labs, when we actually pioneered the use of two-photon to image neurons in brain tissue. So two-photon uses infrared light and enables you to get deeper, not super deep, but at least significantly deep into brain tissue that has the optical properties of a glass of milk. And you can actually see in living brain tissue the structure of these neurons, and you can combine it with calcium imaging. So using calcium imaging with two-photon microscopy, we and many people have imaged the activity of cortical networks in awake-behaving animals. So this is an example of the mouse data. And you can, this mouse is actually looking at the screen where we're stimulating his visual system with gratings, high contrast gratings, which are the same type of stimuli that human and whistle have used traditionally. And simultaneously with an infrared laser, we're taking this two-photon movie on the bottom left, where you can see this is the raw data without any processing. You can see these white cells are the neurons, and you can see them flashing, firing. And this is the analyzed movie on the bottom right. And if you look at this activity, which whenever these neurons turn red, they have at least one action potential. You can see that these neurons like firing together in groups. I don't know if you can appreciate that, but you see that this activity is actually dominated by these systematic little earthquakes, which you can call them attractors, chains, assemblies. We use the word ensemble, which is neutral from the point of view of theory and models. It's just a neutral word, but in my mind, it's all the same. You have a group of neurons that is firing together, and together meaning in the same frame, which could be from 16 milliseconds to 100 milliseconds, more or less. In that little piece of time, boom, you have this bunch of neurons fire together. This is how we analyze the data. We first identify the neurons with a computer program, with an algorithm. For every neuron, we measure the fluorescent intensity as a function of time, as illustrated here. And we transform this into the probability that the neuron had at least one action potential during that frame. And using a conservative threshold of what would be noise, we assemble a raster plot in which every line is a neuron as a function of time. And every one of these black dots is whenever the neuron fires an action potential. In this raster plot, this is a visual representation of the movie like the one you just saw. You can see that there's little lines that run down. So this is this coordinated activity, this ensemble, so group of neurons firing together. You can appreciate it even better if you collapse this raster plot into the histogram of the percentage of neurons that is active as a function of time. And you can see that this histogram is dominated by these little blips, these little earthquakes, when you have approximately 7% of the neurons that happen to be coactive, 7-10%, depending on the experiment. And again, using a conservative threshold of what is real and what is noise, we apply that and extract from these frames the neurons in red that are firing together at a very high probability of occurrence, and we call this an ensemble. So with this type of analysis, we analyze movies like the one you saw. This is the central plot here. This is a raster plot in which the animal is looking at these gratings, and the spikes that are participating in these ensembles are color red. Okay, so you can see that the majority of the activity of the cortex, at least in our lab in these circumstances, is dominated by these ensembles. And the same thing happens if the animal looks at a natural movie. In this case, instead of a grating, we show the animal a BBC documentary in order to capture the spatial temporal dynamics of natural scenes in terms of the bandwidth of the frequencies of spatial and temporal frequencies. And again, the cortex is reacting to this visual stimulus, the primary visual cortex, with these coordinated groups of neurons, ensembles of neurons that are firing together. But look at the top trace here. This is not spontaneous activity. And this happens either in the dark, okay, with no visual input, or when the animal is looking at a gray image, which is not changing, at a static visual input. And if you, in both cases, you see these ensembles, they're still there. In fact, they dominate. With this very conservative criteria, they still dominate the cortical activity. Okay, so spontaneous activity has these groups of neurons firing together already. They don't depend on the visual input. I'm not going to go into the statistics, but just suffice to say that these ensembles repeat. So this is an example of a repetition of an ensemble, but when they repeat, they never repeat exactly. We use, again, different types of statistics, and all of this is very influenced by Moshe, by Evelis. And somehow this cortex has this property in which it never comes back to the exact same state. It's a little bit like a liquid system. No? So you go back to these ensembles, but they're never identical. Some neurons that were there before are not there later. It's a little bit like an image in which every time it occurs in a TV, the image is the same, but the pixels could be a little bit different. In fact, we've demonstrated that individual neurons can participate in more than one ensemble. So this is an example of some ensembles with spontaneous activity in the top, with gratings and visual and natural scenes in the bottom. And these neurons that have these circles around them, this green circle, for example, this one shows up in two different ensembles. The same happens with these neurons here. This is one ensemble that repeats. This is a second ensemble that repeats. This neuron is joining. Neurons can join different ensembles. So it would be like giving it a combinatorial property to this coding. So how do we interpret this data? Well, this is all from looking, okay, and the idea, the hypothesis is that these ensembles are like an intrinsic vocabulary, that the cortex builds these ensembles. This is how it works. And then when you have, and they're there spontaneously, they're always there. And when you stimulate the animal with a visual stimulus, you essentially build a response using these Lego blocks, this intrinsic lexicon, like a vocabulary of words that you put together in order to say a phrase. How do we test this? So for us, our dream experiment is what we call the piano experiment. And this is again influenced by Hopfield. It's the same idea that we discussed at the cafeteria Bell Labs many times. You measure these ensembles, let's say with 2-photon calcium imaging, and this is an example of a movie in which you have these three ensembles, one after the other, and then you go and stimulate these ensembles in the exact same fashion in which you saw them happen. And you can think of this as playing the piano, in which every one of these neurons is like a piano key, and you're essentially pressing like chords in the piano, boom, boom, boom. And then you ask the question, what happens to the system or what happens to the animal? That's how we can test if these ensembles are functional. So one thing is for them to be real, another thing is for them to be good for something. Okay, so how can we test them? with this sort of piano experiment. So for us, the actual keyboard of the piano is something that we call a spatial light modulator, which is a liquid crystal. And this, I credit my student Nikolenko, who came up with this idea. It's a holographic device in which you bring in a collimated 2-photon laser beam. And this liquid crystal has about 1 million pixels that you can control with the computer, and you can change the phase of the light as it goes through the SLM. And by changing the phase, you operate in the Fourier plane, and you can essentially build any pattern that you want in space, in 3D, and in time if you change the phase pattern. And these are examples of some of the patterns that Nikolenko built with 2-photon light. He can write words like Colombia. This is the word he wants to write, and this is the 2-photon version of the word. This is a picture of Causal, and he had nothing better to do than build pictures of Causal and project with 2-photon light and project them back into the brain as if the brain cared. There was a picture of Causal there. Anyway, this is what happens to grad students when the boss is not around. And with this SLM, then we also use an opsin for optogenetics called C1B1. It was also pioneered by a collaborator, Dicera. And this opsin can be used to express it in not in every neuron, but in many neurons in the visual cortex, for example. And then with the SLM, we can activate neurons at will. So this is an example of the worst possible scenario, which you have 2 neurons on top of each other, 20 microns apart, actually 20 microns apart. And in this case, you turn them on both together with the SLM because you build a holographic pattern that stimulates them both at the same time. You can see that here, or you can choose to stimulate only the top one or the bottom one. So you have essentially single cell precision in playing the piano. So we do these experiments with 2 lasers. One is an imaging laser, it's a 2-photon imaging laser, in which you build a 2-photon calcium imaging. We do this normally in 2 or 3D now. And the second laser, we run it through the SLM, and this is our piano. This is how we essentially turn on simultaneously while we're imaging neurons in different spatial temporal patterns. We do this in a wave-behaving mice through the skull, within the skull, again with, in this case, GCAM for the calcium imaging and this opsin for the optogenetics. So the first experiment that we did in this case was Luis Carrillo did, Reed, was a postdoc in the lab, was to play the piano with our elbow, so to speak, okay? This is a difficult experiment. We have to, it's a 2-photon calcium imaging and a 2-photon optogenetic experiment through the skull, okay? So this is not trivial. And he said, okay, let me see if I can actually stimulate all the neurons in the bottom half, in the left half of the field. So he essentially turns them all together, and you can see whenever he wanted to do that, boom, he turned on the laser and all these neurons fire together, bingo. So that means that we can play the piano with our elbow. And then came the discovery, unexpected. He comes to me and said, Rafa, do you remember these neurons that I was firing like with my elbow? I said, yeah, of course, they were very beautiful. You got a very good signal to know. I said, yeah, guess what? They're still firing together. I'm not stimulating them now, and they're firing together. So what happened is that if you stimulate neurons to fire together 50 or 100 times, okay, something happens and they stick together and you build an ensemble. You imprint an artificial ensemble. So this is illustrated here. So this here's a raster plot of the activity of the neurons as a function of time. And you photostimulate them with this, playing the piano with your elbow. And notice how here you start to see in the dotted line, this ensemble that is formed by many of the neurons that you stimulate together that is firing spontaneously. In fact, you bring the animal back to the animal facility, and the next day, these neurons are still firing spontaneously. You've rewired, you've reprogrammed cortical activity by playing it for about 5 minutes. Again, just these 50 to 100 times playing together. And now you build yourself an intrinsic ensemble, an artificial ensemble. Moreover, it gets more interesting than that. So Luis was started to play the piano with a finger now. And this is an example of one of these experiments. Again, he's using an SLM. This is pretty complicated, I have to tell you. 2-photon calcium imaging, 2-photon optogenetics with an SLM through the skull. And he's imaging all these neurons, and he's stimulating this little guy here, this neuron here. And every time he turns the SLM to build this holographic pattern that sends a beam of light only to this neuron, boom, he gets the neuron to fire. Excellent. No? And when he started doing this, playing the piano with the finger, he found that when he stimulated neurons that were part of this artificial ensemble, okay, about 30% of the neurons that he stimulated, once in a while, tripped the entire ensemble. In other words, he discovered pattern completion. So this is an example here. This is an ensemble in pink, and he stimulated neuron 25, which is over here, okay, with the laser. This is the Rafael. And most of the time you stimulate neuron 25 and nothing happens. But once in a while, like here, you stimulate neuron 25 and bingo, all these other neurons fire. Guess which neurons they are the neurons from this artificial ensemble. And this pattern completion happens still a day later. So this is an example of an experiment, in this case, stimulating neuron 19, and this is the ensemble that neuron 19 belongs to. This is an artificially imprinted ensemble. And when he stimulates neuron 19, he can get this ensemble. In this case, this is a different type of analysis. Here we're plotting ensembles instead of neurons. And this ensemble number 1 is this one here, is the artificially imprinted one. And you can see how this neuron 19 is triggering, the good pattern completion cell is triggering this ensemble. He brings the animal overnight, and the next morning he stimulates neuron 19, boom, and he gets this ensemble again. So this pattern completion is very robust. It's actually, it's there the next day, even after 5 minutes of stimulation. So with that, just to turn to the final part of my talk, the final set of experiments, the question becomes, okay, so you see these ensembles, you can actually build them, for God's sake. So forget about statistics, you can actually make them happen with your own hands. So they're really real. The question is, do they matter? And for that, we essentially did this loop experiment in which we measure the behavior of the animal. We measure these ensembles, and then we turn on our piano or SLM, and then we play back these ensembles and see if there's an effect on the behavior. The behavior we choose is a very simple go, no-go task in which the animal has to lick if he sees these vertical bars of light. See, when they happen, the animal licks. I don't know if you can appreciate how the mouse is licking. And we train the animal that if he sees horizontal bars of light like these ones, not to lick. So this is the no-go stimulus. The go is the licking with the vertical bars of light in this case, and no-go is the horizontal bars of light. Simultaneously, we're using two-photon calcium imaging to measure the ensembles that correspond to the go or the no-go stimulus. And the experimental design, it's like this. No? So again, first the vertical goes bars of light, go stimulus. We measure this ensemble. Let's call it ensemble number 1, and the animal licks. Then we stimulate the animal with the no-go stimulus, horizontal bars of light in this case. We measure a different ensemble, okay, and the animal doesn't lick. And then the killer experiment, we turn off the visual stimulus, we turn our piano, our optogenetic holographic stimulation, and we activate these ensembles and see what happens to the behavior of the animal. Let me show you what happens. So first, for technical reasons, it was easier for us to stimulate the no-go ensemble, and this is what happens here. It's a complicated slide, but maybe just focus on this part here, panel E. This is the performance of the animal, the licking. When the animal is trained, so it licks about 80 to 90% of the time correctly. But when we show him the go stimulus and turn on a few neurons from the no-go ensemble, the performance decreases. Now think about it. We're stimulating a few neurons in the middle of the brain of an animal that has 100 million neurons, and we're modulating, we're changing the behavior of the animal significantly. This is an example here.
Alpha question. So what's the minimum number of animals or the minimal number of cells that you need to activate to get those?
We try hard to do with 1, but we can only do it with 2. This is an example of the behavior of the animal. So the white bars are the stimulus. In this case, the go stimulus is the short bar, and the no-go stimulus is the long bar. and the leak is the blue bar. So when we show the animal the go stimulus, most of the time it leaks, not all the time. Here, for whatever reason, it stopped leaking, but here it goes back and leaks again and leaks harder. But then if we turn on our piano and activate the no-go ensemble, you can see how we essentially destroy the leaking in this animal to stop the behavior. Next experiment. Now we're stimulating the go ensemble, okay? But remember, these animals are trained, so they perform close to 100% of the time. So we stimulate the go ensemble. What we did is we lower the contrast of the stimulus. This is one advantage of working with visual cortex, in which we can titrate the behavior of the animal. So we give a stimulus which has very low contrast. It's very hard to detect. In these circumstances, the performance of the animal is very poor. So now he starts not with close to 80, 90%, but more like 50, 60%, low contrast. So with this low contrast stimulus, if we then turn on the go ensemble, look what happens. Suddenly the animal sees the stimulus. So the behavior changes, improves significantly. This is illustrated here with low contrast stimulus. In this case, the long bar is the go stimulus, is the opposite as before. But because it's low contrast, it doesn't matter, the animal doesn't leak. But now if we turn on our piano and activate this go ensemble, look what happens. Every time we show the animal the go stimulus, the animal leaks. Now comes the killer experiment, no stimulus, only piano, okay? So we're activating the go ensemble in the absence of any visual stimulation. And this is an example of such an experiment. Look at the raster plot here. These are the neurons, 9 neurons that participate, are part of the go ensemble. They're illustrated here. And we're activating neurons 9 and 8, which we had previously identified as being suspiciously possibly pattern completion neurons. So with our SLM, again, we build a pattern that turns them on at the same time. And we do that every time there's a blue bar here. And here you can see we turn on the SLM and we get neurons 8 and 9 to fire together. So this happens a few times. Now here, for whatever reason, only neuron 9 fires. This is biology. And most of the time, nothing happens. But look what happens here. We turn on neurons 9 and 8 and we trip the ensemble. Okay. All these neurons that fire are actually part of this ensemble. And when that happens, bingo, the animal goes and leaks. So if we can successfully turn on the ensemble with these pattern completion neurons, the performance of the animal from never leaking goes to essentially almost always leaking. So this is in the absence of visual stimulus again. We're triggering this intrinsic state, and this is generating this sophisticated behavior. By the way, the behavior with these internally generated ensembles is identical in terms of the onset, the speed of the behavior, the number of time of leaks with the normal behavior. The animal cannot tell the difference, whether he's looking at the stimulus or whether we're turning on these pattern completion neurons that trip the go ensemble. Moreover, and this is the last experiment, we noticed in these experiments in 2 animals actually, so this was just a chance to catch it, that in the spontaneous activity of the animal, for whatever reason, the go ensemble turned on. So this is an example here. The dotted lines is when the go ensemble is turning on. And when that happens, again, this is without optogenetics, without visual stimulation, without optogenetics. The animal just sitting there waiting for the next experiment. But when the go ensemble turns on, the animal went ahead and leaked. So the blue bars are the leaking. You can see it here. So again, this is just 2 experiments. This is 2 out of 2. We didn't see it anywhere else. But when we saw it, the animals always leaked. So with this I end. I think the focus of our interest is this emergent properties of cortical circuits. What we see is that the activity of the cortex is organized in these coactive ensembles. You can call them attractors, chains, assemblies, whatever you want, bumps, songs, motifs. They're spontaneous states. Neurons can belong to different ensembles. They can repeat but never identically, and they have pattern completion. Visually evoked ensembles reactivate ensembles that are there spontaneously already. You can see them in the spontaneous activity. So they're intrinsic. Ensembles can be imprinted and recalled artificially from the outside for several days. And in our hands, ensembles are necessary and sufficient for behavior. They're causally related to behavior, to this visual perception task, because if we turn off the go ensemble, we get rid of the behavior, and if we turn it on, we trigger behavior. So this to me is necessary and sufficient. And this is a hypothesis. There's a difference between sensation, which is the way your brain reacts to the sensory stimulus, and perception, which is the idea you have of the sensory stimulus. Because of the fact that we can activate these things intrinsically, and sometimes they activate intrinsically by themselves, like I showed you here. We think that these ensembles are hypotheses that they're internal units of perception, that this is close to what Kant was calling Darstellungen, the idea that we have of the outside world. So it's not just a reflection of the outside world, we can actually activate that internally. I finish with a quote from my mentor, Cindy Brenner, who's argued that progress in science depends on new ideas, new discoveries, new techniques, new discoveries and new ideas from that order. All of this is possible because of new techniques, just like the neuron doctrine was possible because of the new techniques of looking at neurons one by one. And just to give credit to people in particular in the lab, John Miller, who discovered these ensembles in vivo in mouse B1, and Carrillo Reed, who played this optogenetic piano. And also a conflict of interest to declare that our group for Colombia has the patent for this two-photon holographic optogenetics. So we do have a conflict of interest. And with that, I thank you. I wish I was there with you guys in Jerusalem. And I'm sorry I cannot be there now, but it will happen. I will be there, I promise. Thank you.
Thank you, Rafa. We have time for questions, and I'd like to start with the first one. So you showed us at the beginning that even for the icing model and for the Hopfield model, what's really important is the connectivity. Yet you're using patterns of activation, which is an indirect measure of connectivity, and patterns of cluster activation could be explained, for instance, like a common input from different areas. So maybe you can elaborate a little bit on this.
Yes. So we're trying to investigate what is the connectivity in the ensembles. By the way, we know very little about the ensemble. This is very early days. If you think about it, after the 100 years of neuron doctrine, then we need to do 100 years of ensembles now. Where do they come from? What is their anatomy? What is the development? How do they, what is their computational function? How do they relate to these states? How are they affected in disease? So our group and other groups are actually entering these, trying to discover all these things. So I cannot tell you what is the connectivity between these neurons. I cannot even tell you what is the identity of these neurons. We know these neurons I showed you were excitatory neurons, but we're trying to understand what subtype of excitatory neurons they are. You know, there's about 160 different classes of neurons in the mouse primary visual cortex. So, so we know very little about that, what role the inhibitory neurons play. You know? So, but one thing that we're finding, which is very interesting, is that these things are not heavy end, okay? And I have to confess, I'm really embarrassed about this part, because when we discovered the imprinting of these ensembles, this experiment that I showed you, this artificial imprinting, we said, oh my God, this is heck. No, you stimulate the neurons fire together, they wire together. So we wrote a paper, and I have to tell you, this paper was published in Science, so it was not in my little journal. And the conclusion of our paper, the last sentences were saying, and we prove the Hebbian postulate. Okay. Anyway, we started looking for the mechanism of these ensembles, and it's not Hebbian. Okay. So in our hands, this is probably an intrinsic excitability mechanism. If these neurons are firing together, they have to have some sort of connectivity for sure, or be part of some larger ensemble. But what trips them into firing together is not that they build new synapses, it's that they become more excitable. So that means that the same input that you would get from one neuron before now can actually trip your action potential. It's just what Abeles was saying. You're getting closer to that action potential threshold. So the effect is the same. You have a group of neurons that were firing individually before, and now they're firing together, but the mechanism may not be heavy.
So this means that you cannot deduce the connectivity from a single cell to a network just by the pattern of if you stimulate that cell and look at the activity of the cells that are activated by him.
Yeah, I think you probably can induce connectivity. I don't disagree with that, but all I'm saying is that these quick changes that we see again in front of our eyes by activating these neurons, they're probably not due to increases in synaptic strength, but are due to increases in excitability, which effectively is equivalent. So because the strength of the synapse makes the postsynaptic cells fire more frequently. But in this case, the postsynaptic cell fires more frequently, not because the synapse is stronger, but because that cell is actually more excitable.
Okay, thanks. Any more questions?
But Rafa, if the postsynaptic cell becomes more excitable, it is excitable for all the synapses. It's not selective anymore, is it?
Yeah, you're absolutely right. It's not selective, but somehow these neurons that are part of the ensemble are they're flipping together into this state. So I've actually the way we think about it. Again, this is we haven't figured this out yet, no. But let me just share with you this model. Sorry, let me see if I can play. Bear with me. See if I can show you my screen again. So this is what we call an iceberg model. So just imagine this is an ensemble, and there's a bunch of connections that link these neurons, but they're all sub-threshold. You don't see it, but it's there already. Okay, I'm not saying that synaptic strength doesn't matter. I'm saying that it takes time to build these things. And then we turn these neurons on, okay, selectively, only a certain group of neurons. And these neurons become more excitable. So it's like pushing this iceberg out of the water. So this connection that used to be sub-threshold here, now they trigger action potentials. And this is what we see as an ensemble. Yeah. So this is a little bit the model, the sort of iceberg model for ensemble imprint. In this case applies to imprinting. So this is, by the way, I'm just talking only about these ensembles that we can activate artificially, can imprint artificially.
But then you can check it directly. If it becomes more excitable, you can stimulate it with an electrode and see whether it generates more spikes or something like that.
Exactly. This is exactly what we're doing. We're actually in the middle of those experiments, and we're trying to find out what are the mechanisms of this excitability. We think it may have to do with the potassium channel. And of course, the regulation of that could end up having a major effect in the way these networks are actually organize themselves into groups of coactive groups of neurons into this internal state. But what you're saying is exactly the type of experiment that we're doing, to turn them on now with electrodes to see what happens to these IF curves in terms of the impulse response curve of the neuron. And indeed, we're finding that they become more excitable, and this hyperexcitability goes on for a long period of time.
Interesting. Thank you.
Can I have a question?
Yes, of course.
Thank you for the fascinating talk, Rafa.
Thank you.
I wonder if there is some, if you notice some shared properties between the ensemble depending on the stimulus that you present.
Okay, so if you stimulate with a particular orientation, you trip an ensemble that responds to a particular orientation, yeah? And this ensemble is actually better tuned than the neurons themselves. So this is actually one advantage of using ensemble. You could ask, so why does the brain want to use an ensemble? Well, we don't know, but there's one possibility, going back to Lorente, is that this enables the brain to turn these things on internally, so they become independent of the world. Another possibility is that, hey, the group it's better than the individual, so you can do things better in a group. So maybe the coding is more specific. One of the problems with the human and whistle way of thinking and the model is that the tuning of these sensory cells is very poor. I mean, this is one thing that struck me from the beginning. Like, if you build the cortex to detect orientation, how come these cells have such broad orientation tuning, and some will have very narrow orientation tuning? But you would imagine that every different orientation has its own particular ensemble, and this is how the animal recognizes that orientation. So in that sense, there is a specificity between the stimulus and this ensemble. There's a correspondence. Now, there are many more ensembles than we know what to do with now. So this, as you see in these movies, these ensembles are coming up in front of you, right, one after the other. No? So we don't know what all these ensembles are doing, but I can tell you too, in the visual cortex, in the dark, okay, their ensembles are correlated with the behavioral state of the animal, whether the animal is running or not, or whether it was the arousal state. So this is also tells you that we're dealing with a system, I think it's closer to the way Hopfield thought about it, as a neural network where everyone is sort of interconnected and different parts of the brain can participate in computations of other parts.
I meant that if there's some similarities between the stimulus, like if you present a grating bar that it's in a similar orientation, do you see some similar patterns of assemblies?
Yes. So exactly. So what you're asking about the fine tuning, if we change orientation little by little, what type of ensemble we get? So we've done this, but not with many orientations. We've maybe done it with 8 or 16 orientations, and we do get, as I'm telling you, 8 or 16 different ensembles that correspond to different orientations. But we haven't really looked into the. I think what you're asking, what happens if you change the orientation a few degrees? Do you get a different ensemble? And this is actually it's a great question. It's like you get another attractor, or is it still part of the same attractor? Yeah, I don't know. We don't know.
Thank you.
Yeah.
More questions?
I have a question. I guess I'll go. So I want to know about the size of these ensembles, because presumably the ensembles are not just the ones that you see in the field of your microscope, right? There are probably a bunch of other neurons that are outside of the field that are also responding. So how big do you think these are? And do you find it a bit surprising that with just the neurons that you see within the field of the microscope, you can generate behavior?
This is a great question. First of all, not only I found it surprising, I didn't want to believe it when Luis said, I'm going to stimulate this Go ensemble and change the behavior of the animal. And I told him, it will never work. This is you stimulating 2 neurons in 100 million. This will never work. In fact, I used to be super critical of the experiments that were done by Michael Brecht, stimulating 1 neuron in motor cortex in a rat and changing the whisker behavior. No? And that's it kind of true. No? Anyway, it works. It's robust. And just in case you don't believe it, these results were replicated by the Dicerot lab last year as well. So this is now at least 2 labs in the world that can change the behavior of the animal by stimulating 2 neurons. We cannot do it with 1, okay? But we can do it with 2.
Also in the visual cortex? Also in the visual cortex?
Exactly. The paper, the Dicerot paper was first author Marshall, was published in Science. Our paper, first author Carrier Reed, was published in Cell. Completely consistent with each other. No, same result, different method, different approach, different theory, but the same result. You stimulate a couple of neurons, you trip the ensemble, you generate the perceptual state, the animal goes and leaks. So I just couldn't believe when that happened, no? And that's when ABLS comes in. So how could that work? Well, imagine this is pattern completion, except you're doing pattern completion. With pattern completion, you can trip the first ensemble. But this ensemble has a bunch of neurons connected to other ensembles around the brain. And if by chance this one of these neurons is connected to a pattern completion neuron of the second ensemble, it trips that. And that way you can get like a critical phenomenon where you have essentially an avalanche of ensembles throughout the brain. And one of those ensembles could be in the 12th cranial nerve nucleus that controls the tongue. So that's how I could explain conceptually that stimulating two neurons in the back of the cortex can generate this behavior in the mouse. But back to your first question, we don't know. These ensembles, we're only looking at a little corner of the TV screen. You could criticize me and say, well, this is the same criticism that you made to Hopfield. Well, how about the rest of the TV screen? We don't know. And that's the reason why we're trying to build, we as a community, better methods to try to capture completely the activity of nervous system so that we can actually finally put that question to rest. And I just wanted to show you maybe an example of our work in Hydra. So David mentioned that we also work in Hydra. Why do we work in Hydra? Precisely for that reason. So I'm going to show you a movie in which this is probably the first time that you can look at every action potential from every neuron in an animal. Okay, so this is a little cnidarian, transparent, a couple of millimeters tall. These are calcium imaging from about 95% of the neurons in the animal. And in some parts of the movie, the signal to noise is good enough that we can put our hands in the fire that we're catching every action potential, okay? So this is the beginning of looking at the whole TV screen. So just like we did this work in Hydra, published it 3 years ago, people are doing similar work in the worm. They're not getting yet to every neuron, but they're getting close. No? And in the larva of the fly and in other systems. So little by little, as it happened with the human genome, we'll be able to catch this entire TV screen. And there you can actually, I can answer your question. Say, how big is the ensemble? I can tell you, in this case, you're looking at I think there's about 7 ensembles in front of you here. And how many neurons do they have? We can tell you exactly what number of neurons that they have. And then we can smile and you can say, oh yeah, but maybe there's a neuron that you're not recording from that is part of the ensemble. And then I can smile and tell you there are no more neurons, that's it. we're seeing every single neuron. So this is the beauty of this approach where you can actually capture every spike that you've imaged every neuron. So that all the ensembles, in a way, you sort of completed the experimental approach to the system. Now, of course, the challenge is to decipher what it means.
So for the sake of balance, there are some questions which were raised on the chat. So I'll read a couple of them. So Jonathan Canbon is asking, how many spikes are elicited during a single stimulus of the neuron? And how much stronger is the artificial activation relative to activation of natural stimuli? Okay.
Going back to the mouse data now, we trigger a burst of spikes, okay? And when we do optogenetics, we calibrate the optogenetics so that on average, the activation with the optogenetic piano is similar to this endogenous activation. Okay, so you should think of burst of spikes. Do not think of individual spikes. Here for the mouse data, for the Hydra data, then I can tell you we're looking at single spike. No, mouse data is just burst of spikes. So yeah, so when I said I would stimulate neurons 100 times together, and by the way, these are experiments that were consistent with old work that was done in Israel, actually, part of the Avalos group, also doing heavy and imprinting, so to speak, imprinting of firing neurons together and changing the correlation coefficient between these neurons in vivo in monkeys. Now, this is all consistent with the same idea. No? But in this case, in our case, actually, we're stimulating these neurons with bursts of spikes.
Okay, and another question from the chat. Itzik Norman is asking, how specific is it to the visual cortex?
Okay, so we don't know. I think we're early days. We're starting to explore this in hippocampus as well, this imprinting of ensembles, but I think it's consistent with what people have been describing in the hippocampal system. People talk about engrams in the field of memory. What they mean by an engram, I think it's the same thing as what we mean by an ensemble. And they talk about attractors in hippocampus, and the cortex people are a bit more reserved, but I think it's very consistent with the literature at this hippocampus, but I cannot tell you. And from our own data, we've only looked at V1 and a little bit at prefrontal. Oh, sorry, I should say it happens also in auditory cortex. So we've seen the similar things in A1 and in different auditory areas in the mouth.
Today, may I have a comment?
Yes, of course.
May I comment?
Yes.
Go for it.
Okay, so asking about the different areas, as you know, we work in the motor cortex, and in the motor cortex, we used another way to imprint a new assembly, new group, by conditioning one neuron to increase firing rate and recording all the others. So the conditioning is there are no electrical or optical stimuli. There is only conditioning. If this neuron is active, the monkey gets reward, and then a new pattern of activity evokes in the population that we record. So, so this is also in complete agreement with what you described today.
Yes, Ellen. When I mentioned that there was consistent with experiments done in Israel, I had your older paper in mind, which was very influential. There's been many years.
Yeah, there is a new one not accepted yet. You'll see the new one. It's closer.
Yeah, but it's very consistent. I think the what we're seeing, it's one of the first people to see something like this, no? But again, it's all consistent with the possibility that the cortex is very plastic and you can actually reward these things on the fly. And again, at least from our preliminary data, part of this rewarding may have to do with intrinsic excitability in terms of the actual biophysical mechanism.
Okay, so one last question before we finish.
Yes, I would like to do a question.
Go ahead.
Is it known what makes some ensembles more stable than others, or is every ensemble equally stable?
Yes, this is a great question. We actually have a paper in bioRxiv that we're working on. It's been reviewed precisely on this issue, on the stability of ensembles. So, some ensembles can last for at least 46 days in V1. So this is the longest we've measured them. Okay. So we know that some ensembles are stable at least for 46 days now. And in those stable ensembles, there's some neurons that are actually very stable. So those you could call core cells. What are these neurons? We don't know. They seem to have stronger functional connectivity with other neurons. If you use graph theory to analyze their conditional probability of firing with respect to the other neurons, these neurons have higher ranks in terms of the number of functional connections with the other neurons. But but this could suggest that there's some, and maybe those are the pattern completion neurons, that there could be some within these ensembles. Not all neurons are the same, and there's some neurons which maybe because of chance, or maybe because they belong to a different subtype, or maybe because they have stronger connections, dominate the activity of these ensembles.
Okay, so for the sake of time, we have to stop here. I'd like to thank Rafael Yuste for his fascinating talk, and welcome you all to the next week's seminar. And thank you again, Rafa.
Thank you, and warm greetings again from New York. Shalom to everyone.